Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Maximizing PK and ADA Evaluation Efficiency Using Cutting-Edge Rapid Antibody Development and Technology. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank GenScript, who developed the content for this presentation. GenScript is the leading contract research organization in the world, providing gene, peptide, protein, CRISPR, and antibody. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services, encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Now, it's my pleasure to introduce our speaker for today's event, and she is Dr. Wei-Fong Tan. She is the Global Product Manager who supports GenScript's antibody production service currently. She graduated with her PhD in Biological Sciences from Nanyang Technological University in Singapore, and continued to work as a postdoctoral research fellow in cancer research for another 2 years. After she joined GenScript, Dr. Tan has supported the proteins and antibody production services in APAC region for a year. She provides application support for pre- and post-sales activities for services, as well as updates and seminars to support co-stakeholders, distributors, and customers. Currently, she is supporting new service launches and training in antibody production services. Now that I've got the introduction out of the way, it is now my pleasure to pass the mic and the controls over to Wei-Fong. So Wei-Fong, when you're ready, you may begin.
Sure. Thanks, Sonia, for the introductions. Hi, good day, everyone. Thanks for taking time to attend today's webinar. And it is my pleasure to be here to share how to maximize KPK and ADA evaluations efficiency with our cutting-edge rapid antibody development technology. I'm Hui Fun, the Global Product Manager from GenScript. Please feel free to leave your question in the chat box, and I will try my best to reply you after the talk. In today's webinar, I will first talk about what is anti-idiotype antibody and its applications. After that, I'm going to share why rabbits are preferred hosts for generating high-performance anti-ID antibody. Follow with our GenScript rabbit anti-ID antibody discovery strategies and capabilities, and some success cases project we have delivered. Lastly, I will end my presentation with our polyclonal antibody ADA discovery strategy and some case study. First, let's start with the overviews of anti-idiotypes antibody. So what is anti-idiotypic antibodies? Anti-idiotypics are also called anti-ID antibody, which are antibody that bind to the idiotypes of another antibody, usually an antibody drug. An idiotype can be defined as the specific combinations of idiotype present within an antibody's complement determining regions. A single idiotype is a specific region within an antibody variable regions which bind to the paratopes of the different antibody. The paratope could be bind directly with the epitopes of the antigens. Therefore, an idiotype can be considered almost synonymous with an antigenic determinant of an antibody. There are three main types of anti-ID antibody based on the way they interact and detect on antibody drug, which shown here. The first, which is the antigen blocking anti-ID antibody, is named so because the antibody drug paratopes and idiotopes overlap with one another. Because of this, the antibody drug targets antigens and the anti-ID antibody will compete with one another, and anti-ID act as an inhibitory and neutralizing. Therefore, this form of anti-ID antibody will only detect the free antibody drug. The second type of anti-ID antibody is called non-blocking because the antibody drugs, paratopes and idiotopes do not overlap. Therefore, the anti-ID antibodies and the antigens can simultaneously bind to the antibody drug without affecting one another binding capability. Because of this, non-blocking anti-ID antibodies are used to detect all forms of available antibody drug, which includes free and antigen bound. The third type of anti-ID antibody is a complex specific anti-ID. This is because the anti-ID antibody cannot bind to the antibody drug unless the drug is already bound to its antigen. Therefore, this type of anti-ID antibody is only able to detect bound antibody drug. The blocking and non-blocking anti-ID antibody are useful for PK or ADA kit assay development. Anti-idiotypes antibody can be generated to bind specifically to a biological drug. This highly specialized antibody can be used to set up pharmacokinetic assay in different formats to measure free or total drug level in preclinical and clinical samples, or it can be also used as a positive control in ADA assay. Since most of the anti-ID antibodies are generated against a specific biological drug, they are commonly used in preclinical practice for pharmacokinetic analysis. PK is the studies of drug metabolism throughout the body. Specifically, clinicians will determine the rate of drug absorption, distribution, bioavailability, and excrete in various cohort of patients. In order to accomplish this, researchers need to be able to track antibody drugs which are bound or unbound to their designated targets at various time points post-deliver. Through the use of anti-ID, various forms of therapeutics can be easily tracked and quantified in patient serum, blood, urine, or other body fluid. In the right figures, the blood drug concentration time curve, we can see that the durations of the effective drug concentration is different in the blood because of the different administrations methods. So a quantitative method for detections of the effective drug concentration and the toxic drug concentration is necessary for the assay. Immunogenicity is the ability of a therapeutic such as antibody drug to induce a humoral or cell-mediated immune response, which will lead to developments of anti-drug antibodies. Anti-drug antibodies lead to the negations of all antibody-drug related effect, essentially completely inhibiting the therapeutic aspects of the drug. Some unexpected immune reactions may lead to neutralizing biological activity of drug or cross-reactive immune reactions. These may lead to the allergic reactions, arteries or vein blockage, and cytokine release symptom. The non-blocking ADA could bind to antibody drug and affect the drug ADME, while the blocking ADA could affect the binding of the drug to the targets, which may reduce the drug efficiency. Therefore, it is extremely important for researchers to analyze the immunogenicity of a new antibody drug during the preclinical analysis An anti-drug antibody is very similar to an anti-ID since both bind to the same antibody drug. Therefore, a pool of polyclonal anti-IDs can be used as a positive controls when analyzing for the presence of the anti-drug antibodies within the patient sample. Next, I will share what's so special about our rabbits generated antibody and why rabbits are the preferred host for generating high performance anti-ID antibodies. In the market, there are different antibody discovery strategies, for example, through traditional hybridoma platform, display library, B-cell cloning, and so on. In GenScript, our rabbit's antibody discovery platform is performed via traditional hybridoma technology. Our rabbit's antibody discovery is performed via B-cell cloning platform or by using a beacon single B-cell screening platform. Rabbit's antibody discovery platform are always highly recommended to customer due to the rabbit's generated antibody have higher affinity, specificity, and diversity. I will share more information in the following slides. Antibody drug discovery is an arduous process that often starts with the extensive screening of antibody library to identify optimal candidates, namely those having high affinity antigens binding and others desirable functions properties. The process for researching and developing new medicine is growing in difficult and in length. On average, it takes at least 10 years for a new medicine to complete the journey from initial discovery to marketplace, with a clinical trial alone, which is taking 6 to 7 years on average. The average cost to research and development each successful drug is estimated to be 2.6 billion. This number incorporates the cost of failure, which include thousands and sometimes millions of compounds that may be screened and assessed early in the R&D process, only few of which will ultimately receive approval. The overall probabilities of clinical success is estimated to be less than 12%. Success require immense resources, which include the best scientific mind, highly sophisticated technologies, ever-evolving manufacturing process, and complex project management. There are some of the challenges which scientists encounter nowadays on the antibody drug discovery route. Antibody are expensive to produce because of the difficult process of isolating and quality testing thousands and thousands of candidates. It sometimes is also limited by the funding issue as well. This process can also take up to years and require a lot of technical expertise to synthesize one therapeutic drug. Therefore, timeline and the financial issues are always the main challenge in drug discovery and development. Other than that, manufacturing issues such as instability of antibody, undesirable byproducts from the manufacturing process, yield and quality inconsistent or unable to reproduce products also will lead to discovery process delay. Appropriate target screenings and selections methods are required to ensure the targets and leads are correctly selected before further validations. Insufficient validations of drug targets at an early stage has been linked to costly clinical failure and low drug approval rate. Besides, antibody drug developability, safety, and efficacy play a crucial role in antibody drug discovery. This involves several optimization strategies and methods to improve the final drug candidates. Without a proper drug optimization and modification step, many potential candidates are unable to pass through the safety and efficacy tests. Besides, lack of PK or ADA development capability will also affect the efficiency of safety validations. Drug safety and efficacy validation is crucial in the drug discovery process. pharmacokinetic and immunogenicity research run through the entire life cycle of the drug developments. More attention has been paid in therapeutic research, which has become the consensus of the drug administration authorities and medical research. Without passing through the authority requirements, the selected drug candidate will not market and all the hard work will go into drain. Therefore, the establishment of non-clinical analysis and evaluation method can provide early guidelines for clinical research. It helps to improve the safety and efficiency of the drug. The FDA, MMPA, and others regulatory agency require the drug's effectiveness and safety must be demonstrated in animals before it enter the clinic. Both preclinical and clinical study need to study the PK and ADA of the drug. Therefore, the establishment of a good PK or ADA assay is extremely important for the preclinical, clinical analysis and evaluations of the drug. Like mentioned in the previous section, NTID is used to set up pharmacokinetic and ADA assay. Making a good quality NTID will improve the drug efficacy and safety evaluations. In the NTID developments, there are challenges in the developing good quality NTID antibodies. Low immunogenicity of the antibody targets CDR regions. This will cause low immune response and therefore low NTID will be generated. Besides, we commonly see the high cross-reactivity with humans IgG occur which recognize FC region, therefore low percentage of positive clones were obtained in the anti-serum. Lastly, generations of specific target clone is a tedious task as well. Some assay require blocking anti-ID and some require non-blocking. Therefore, multiple rounds of assay validations are needed to achieve suitable clones for the assay. Therefore, finding a solution to overcome the challenge mentioned just now, which will largely help customers to reduce the quality concern for their anti-ID developments. For the challenge which I mentioned previously can be solved by the following solutions in mouse hosts through performing immunogen design with KLH conjugates or using FAB2 as immunogen to reduce the non-specificity of FC-generated anti-ID or adjust the immunization schedule to improve the immunogenicity of the targets in anti-ID developments. However, going through all these strategies to improve the anti-ID developments in mouse if still unable to develop a good quality antibody, which you should consider to switch to another host like rabbits, which will actually effectively increase the success rate of getting a good one. Or this strategy can be also applied on rabbit as well to improve the development strategies. Rabbit generate high affinity, specificity, and diversify antibody, which can provide a better antibody to recognize more targets epitopes for anti-ID developments. On average, rabbit's monoclonal antibody has highest sensitivity and specificity compared to this, and immunized rabbits' spleen contain as much as 50 times more lymphocyte than mouse spleen, which provide numerous sources of B cell for generating a large B cell pool for the selections of higher affinity and specificity antibody clones. Beside from the Western blot result here, you can see the immunized rabbit serum can detect more epitopes compared to the mouse immunized serum. This phenomenon may be caused by reduced immunodominance in the rabbits, which allow the recognitions of low abundant proteins or epitope that usually are not visible to the mouse immune system. Here is a summary slide of rabbit's antibody advantages. Beyond its large body size, which will actually facilitate large collections of antiserum and immune cells, and the rabbit's antibody are able to recognize epitopes on humans' antigens that are not immunogenic in rodents, increasing the total numbers of targetable epitopes and facilitate the generations of antibody that cross-react with mouse autologs of humans. Other than that, due to rabbits' limited inbreeding allowed for wide antigenic recognitions, and rabbits have a unique IgG features which contains only one subtype of IgG, more disulfide bonds, greater variations in length and sequence, especially in CDR3 regions. And rabbits elicit strong immune response against small molecules and heptine, which is uncommon in rodents. In summary, rabbits have a robust and unique immune system that enable the generations antibodies of superior specificity, affinity, and diversify compared to rodents. Here is a comparison table show the differences between mouse and rabbit generated anti-ID. As mentioned in the previous slide, rabbit generated anti-ID can be recognize more epitopes and have a higher affinity and more diversified antibody generated compared to the mouse platform. Rabbits have a strong immune response to non-proteins antigens, and mouse discover drug candidates. Furthermore, our GeneScript rabbits antibody anti-ID package, we can provide guaranteed blockers and non-blockers anti-ID package. So why not choose a better package with a better performance antibodies? And Sonia, there is a poll question now.
Yes, and I'm going to jump in right now and launch our first poll question. And there you go, audience members. You should see that blue screen take over. Now, this is done in real time, so your participation is strongly encouraged and very much appreciated. So the first question that we have for you is, what are the benefits of rabbit-generated antibodies? So please go ahead and make your selection. Is it high affinity, more diversified, high specificity, or all of the above? And WePhone will give us the correct answer as soon as we get everyone participating in this poll question. So just click on any of the above there, or below, I should say. Okay, that looks like it's the majority of our audience, but I want to leave it open for a few more seconds just to get a few more people in here to participate. And okay, that looks like it's everyone. Perfect. Thank you so much. Now I'm going to close the polls and let's take a look at the results. So here are the results. We have here 0% for high affinity, more diversified or high specificity, 0%, but 100%, everyone in our audience chose all of the above. So what is the correct answer?
Thank you. So the answer is all of above, and the audience all answer correctly. So
Yes.
like I mentioned, like I mentioned previously in the previous slide, and rabbit-generated antibody actually have a high affinity, more diversified antibody, and higher specificity compared to the rodents one. Okay.
Okay, thank you.
Thank you. So in the next sections, I will talk about what are our GeneScript rabbits anti-ID antibody discovery strategies and capabilities. In GeneScript, our rabbit's antibody discovery is performed by a B-cell cloning or Beacon single B-cell screening platform. In this rabbit's monoclonal discovery workflow, customer can select our proprietary mono-express fast immunization strategies to shorten the turnaround time from conventional immunization, which requires 6 weeks, and the express immunization just require 4 weeks to complete the immunization part, and the immunized serum titer is not affected by this short turnaround time. Besides, we isolated the PBMC and without performing cell fusion, which will actually avoid B cell loss due to the fusion efficiency. We also perform fax-free methods, antigen panning enrichment to collect those immunogen positive B cells to further culture, which not only increase the cell variability, but also increase the positive cloning screening afterwards. Besides, we can also perform various screening methods on the B cell supernatant we have isolated and culture for the example like ELISA, FACS, Western blot, IHC, staining to validate whether they are actually positive towards the targets and suitable for the certain applications. Since multiple rounds of optimization and efforts have been made in our B-cell cloning platform since 2020, our R&D teams have tried out different improvement measurements, for example, like antigen-positive B-cell enrichments, plating method optimization, PBMC isolation optimization, and streamlined productions workflow, which you can see from the numbers of seeded plate needed to screen the positive clone in the green bar here are actually reducing from year to year. Even we cut down the seeded plate number, but we obtain more positive clones from the fewer 96 well plate seeded from this blue line. The project one-time deliver success rate is increasing and achieve more than 92% at year 2023, which means that there are minimum numbers of failure orders. You can also observe that the range of the lesser numbers of positive clone deliver become lesser from year to year. And then beside B-cell cloning can provide more than 1,000 antigens positive clones for further screening in year 2023. Positive clones deliver to meet all the screening condition for one round of experiment to get positive clones of 20 to 50 clones is increasing from 2021 to 2023. Besides, so far on our success project delivery rate is more than 98%. With all the improvement and capabilities of our rabbit's antibody discovery platform, we are able to shorten the discovery timeline to 9 weeks, as we know that a rabbit-generated monoclonal has better performance compared to mouse, but rabbit's monoclonal antibody pricing is always more expensive than mouse in the market. However, there is a big news that is now we are able to match our rabbit's anti-ID pricing as our mouse pricing. So when both price are starting from the same, why not choose the better one? We offer the cheapest and faster rabbit's anti-ID in the markets. In the first step of the anti-ID development, which is the immunogen preparations, we can actually perform the antibody fragmentations and conjugation in this step. For the Fc regions of the antibody drug is the same as the antibody circulating in the human body. Therefore, the antibody fragmentation is used to remove the Fc part from the antibody drug by propane or pepsin digestion, and afterwards go through the protein A or protein G purification to remove the Fc region to avoid the non-specific developments of anti-ID. Afterwards, the FAB or FAB2 can be used for immunizations. Other immunogens such as like bispecific, VHH, SCFE, and antibody drug conjugates can be directly used for immunization. However, for small molecules, peptide, DNA, and RNA require conjugation. The inactive virus and virus-like particles could also be used directly for immunizations. Others, like GCT-related element like CAR-T, TCR, Cas9 can be used as immunogen directly, while the siRNA, sgRNA, DNA, and RNA require conjugation before immunization. Here, I would like to share one of the case study from a CAR-T development customer. They would like to develop an anti-ID using a SCFB as an immunogen for their CAR-T. The customer first try out with another company to develop this anti-ID in the mouse platform. However, no available good clones were generated after 20 mice were immunized by them. Money and the time are actually wasted in their first attempt. And this customer come to us, and we suggested to try out with rabbits host, as we know that rabbits have a robust immune system, which can generate high specificity, affinity, and more diversified antibody. The goal of the projects is for FACS application. So SCFB was immunized in the rabbits with express immunization. After the rounds of immunization, the serum titer is good, and then we move on for further B cell cloning. After B cell cloning, we are able to get 192 clones were positive towards to SDFV hits and negative to the control. And we selected the top 97 clones for further verifications of overexpressed cell line, and ultimately customer selected 34 clones from the overexpressed cell line FACS data. These are some of the representative FACS data. You can see that clone 1, clone 2 supernatants are able to detect the overexpressed cell line, and we successfully deliver FACS positive clones for customer application. In the next comparison case study, customer would like to actually generate 4 blocking anti-ID and at least 1 non-blocking anti-ID from its tri-specific antibodies. We try out this project on the mouse immunization and rabbits immunization simultaneously, and the SESB his was used as an immunogen and then to perform the immunization. After third rounds of the immunization. Here is the indirect ELISA result of the immunized serum of both species. We can clearly see that the immunized serum from both species have higher titer with the immunogen targets compared to the human IgG or control IgG1. Besides, we also noticed that the immunized rabbit serum have a higher OD level compared to mouse immunized serum, which suggests that rabbit elicits strong immune response. In this project, multiple counter screenings were involved, which make the project more tedious. After proceeding with the hybridoma culture from mouse, only 27 clones are SCFV hits positive and also weak cross-reactivity to others IgG control. Therefore, no good clones were selected. From the rabbit B-cell cloning strategy, we are able to discover 133 clones of SCFV hits positive and negative with others control. Therefore, the positive clone numbers are higher from the rabbit generated platform compared to the mouse. We move on further for blocking ELISA AC, and the top 96 clones are blocker and have the blocking rate more than 30%. Ultimately, customers selected the top 3 clones from these for antibody productions and further validation. This is the result of the blocking ELISA from the purified rabbit's anti-ID. In conclusions, rabbit generated higher positive clones numbers in anti-ID discovery. In the last case study for the small molecules ADC drug targets in rabbit's monoclonal anti-ID, the goal is to develop a MMAE antibody not recognizing MMAF. The MMAF different from the MMAE in that it has a charged phenylalanine at the C terminal, while the MMAE is uncharged. Small molecule MMAE was used as a immunogen, and MMAF was used as a negative screening material. We are able to generate 214 clones which are positive to ADC and negative to the linker with KLH. We selected top 192 clones for further validation by blocking ELISA, and 24 clones are positive to MMAE and negative to MNAF. Ultimately, we express 15 clones and validate with ELISA assay against, and all are positive in the ELISA. This is a summarized figures of our deliver anti-ID projects. We can see that the mouse-generated anti-ID have a higher percentage of 1 to 50 positive clones delivered after positive and negative screening. However, the rabbits generated anti-ID have a larger percentage of range 101 to 500 or more positive clone delivered. The average positive clones from rabbit-generated anti-ID is around 713 clones compared to the mouse, which is 204 clones. So more positive clone available from the rabbit's anti-ID provide a better chance for customers to select a better performance antibodies. We are the award-winning custom antibody provider with over 20 years of experience and deliver nearly 200 success antibody projects to date. Highlight of our NTID services, we are able to deliver about 90% success rate in NTID project and 95% success rate for blocking, and for the non-blocking, which is about 90% success rate. And we are able to provide a guarantee blocking and non-blocking rabbits monoclonal NTID package. Besides, as I mentioned earlier, we can accept various antigen type for immunization. Our rabbits generated antibody has more diversified clone number compared to the mouse. Our rabbits NTID projects are 3.5 times more positive clone numbers discovered compared to the mouse NTID. Our turnaround time is faster in the market for rabbits NTID discovery, which is as fast as 9 weeks. Hi Sonia, there is another poll questions.
Okay, and I'm going to launch our second poll question, and there you go, audience members. What are the benefits of GenScript Rabbit Anti-ID MAB? So please go ahead and make your selection. Is it fast turnaround time, as fast as 9 weeks, guaranteed blockers and non-blockers package, higher positive clones delivered, or all of the above? Okay, and if we can get everyone participating in this poll question, it'd be greatly appreciated. Okay, I'm going to close the polls in a few seconds. I'm just giving a couple more members in our audience a chance to make their selection. Okay, and that looks like it's a majority now. Okay, perfect. I'm now going to close the polls. Let's take a look at the results. And here are the results. There we go. And 100% of our audience chose all of the above. So,
Okay.
WePhone, is that the correct answer? I hope so.
Yeah, yeah, this is the correct answer. So yeah, we for our GenScript Rabbits anti-ID monoclonal package, we are actually the faster in markets, which is as fast as 9 weeks, and we do provide a guarantee blockers and non-blocker package as well. And based on what we have delivered so far, our Rabbits anti-ID have a higher positive clone level compared to the mouse. Thank you, Sonia.
Wow. Thank you.
Okay, so let's move on. In the last section, I will end my presentation with GenScript polyclonals ADA discovery strategies and capability. Like mentioned earlier, we are able to accept various antigen target. If it is a drug antibody with SC, then we are able to perform a fragmentations and immunize the FAB2 part. Once the immunogen is prepared, we will perform either express or conventional immunization in rabbits, depends on the customer project requirements. In the ADA polyclonal antibody developments, antibody is isolated from the immunized rabbit's blood, and the ADA polyclonals will be purified with the antigens affinity columns and further purified with total human IgG column to remove the cross-reacting ADA. It takes around 7 weeks to complete the project and get the ADA polyclonals with different guarantee amount depends on the customer needs. I would like to share some of the ADA polyclonal case study here. In standard monoclonal targets project, monoclonal antibody is digested by pepsin, and then FAB2 is isolated for immunization. In this project, we perform the express immunization. From this plot, we can find out that purified ADA is positive towards to the monoclonal antibody, but negative to the total human IgG. We successfully delivered 26 milligram to client end in 73 business day. In the following ADA case study, which are using the ADC or protein drug conjugate immunogen, in the ADC target project, the generated ADA is to the target immunogen and negative to the total human IgG. About 23.2 milligram total ADA was successfully delivered to client end in 40 business day. And in the paylated protein project, the generated ADA is positive towards the target immunogens, and 75.4 milligram total ADA were also successfully delivered to client end in 60 business day. In the following ADA case study, which are using a peptide, semaglutide is the target, which is quite popular nowadays. It is used for treatment for type 2 diabetes and anti-obesity medication used for long-term weight management. It is also a peptide similar to the hormone glucagon-like peptide 1, modified with a site change. In this semaglutide target project, the generated ADA is positive towards the target immunogen. About 42.7 milligram total ADA was successfully delivered to client and in 90 business days. In this case study, which are using a siRNA or payload immunogens, in the siRNA targets, the generated ADA is positive towards to the target immunogens. About 3.5 milligram total ADA was successfully delivered to client and in 45 business days. In the payload project, the generated ADA is positive towards to the target immunogen. About 1.5 milligram total ADA were also successfully delivered in 90 business days. Here is some ADA development summary. Based on the different immunization strategy, and the express immunization strategy doesn't affect the ADA amount generated with a shorter immunization timeline. And then other than that, although the ADA generated from VHH is lower than the monoclonal antibody as a target immunogen, we are still able to generate ADA amount more than 1 mg per rabbit. Moreover, our generated purity is more than 90%, and the ELISA titers of purified ADA is more than 1 million. The cross absorption is less than 2%. Here is the ADA development cases we have delivered so far. We actually accept various antigen targets, which included standard IgG or bispecific or peptide, small molecule, and so on, which I have shared previously. And we have successfully delivered to date is about 69% of the antibody drug, 10% of the payloaded proteins, 9% of peptide, and then payload, which is 7%, and then the sRNA, which is about 5%. Sonia, there is the third poll questions.
Okay, and I'm going to launch it right now, and there we go. What kind of immunogen targets could GenScript accept for anti-ID development? So please go ahead and select from the below. Is it siRNA, ADC, VHA, or is it all of the above? And I'll leave the polls open for a few more seconds to get everyone involved in our final, not our final, we have one more poll question after this one. Okay, perfect. That looks like it's the majority of our audience that have participated in this third poll question. So I'm now going to close the polls. Okay, thank you very much. Here we go. Okay, and here we go again. 100% of our audience chose all of the above. So, WePhone, what's the
Yeah.
answer?
The answer is all of
Okay,
above. And because like what I mentioned, we are able to accept various kind of target antigens. So not only just standard IgG, we are able to accept like sRNA, ADC, or VHH bispecific as well. Thank you, Sonia.
fantastic. Okay.
Yeah. So okay. So in summary, our rabbits monoclonal anti-ID discovery platform enable researchers to generate a high-performance antibody as fast as 9 weeks. We also can offer the guarantee blockers and non-blocker anti-ID package. For the ADA polyclonal, we are able to generate as fast as 7 weeks with a guarantee amount. The ELISA titers is bigger than 729K, and cross rate is less than 2%. So that is the last poll question for Sonia.
Okay, yep, and I'm going to launch it now, and here we go. Last poll question, everyone. Are you going to work on anti-ID? Sorry about that. Are you going to work on anti-ID projects in the next 3 months, in 6 months, not decided, in a year or more than a year? Please go ahead and make your selection. Okay, and now we're just getting everyone joining in on the final poll. So here we go. And I'm going to leave it open for a few more seconds, WePhone, because we're just getting everyone involved in this now.
Yeah, sure.
Okay. All right, that looks like it's the majority of our audience. Thank you very much for participating in our final poll question. Now I'm going to close the polls and let's take a look at the results. So we have in our audience, oh, actually, I'm not going to show the results right now because we have to jump into the
Last one.
Q&A portion of the webinar.
Yeah.
Okay,
That's all for my
perfect.
presentation today, and thanks everyone for their attention. And please feel free to leave your question in the chat box. Yeah, Sonia, you can take the floor. Thank you.
Okay, thank you very much. And I will take over the controls here, and then we will begin the Q&A portion of the webinar. So we'll put on our webcams for that, and I'll invite WePhone to put on her webcam.
Okay, sure.
Okay,
Hold on.
and then just as a reminder, just like WePhone said, we are going to do the Q&A portion of the webinar. So if you're attending via GoToWebinar, please use the questions window. And for those of you watching on LinkedIn Live events, feel free to post your questions in the comments section. Okay, so while you were speaking, I did receive a bunch of questions. So let's start with this first one here. This audience member is asking, what are the benefits of using GenScript Rapid anti-ID discovery platforms?
Okay, cool.
That's a great
Thanks
question.
for the questions. And like I mentioned, we are able to accept various kind of antigens. Therefore, as mentioned earlier, also rabbit can actually generate a strong immune response to the small molecules or heptene. So, which means that rabbit actually able to generate more positive clone for that. Besides that, we also utilize a B cell cloning platform, which is our proprietary B cell enrichment methods. We actually increase the positive B cell and then not affecting the B cell variability. And then through multiple rounds of the improvement, we are able to shorten the turnaround time and then increase the positive clones numbers. At the same time, we are actually also increase the success rate as well. So we are confident to deliver what customer want. And then beside that based on our case study, and we also noticed that rabbit generated anti-ID are able to provide more positive clone compared to mouse. Therefore, customer can select more different kind of varieties antibody for their assay. And lastly, as we know that rabbits actually have a higher affinity and more diversified antibody and higher specificity. Therefore, if when the pricing of the mouse and rabbit are the same, why not choose a better one? Yeah,
Yeah, yes. Thank you so much for that. Okay, we'll
no
go to the
problem.
next question now. This audience member is asking, what's the success rate for blocking and non-blocking antibody and anti-ID projects? That's a great
Okay.
question.
Yeah, that's a great question. Like I mentioned just now, the
Mhm.
success rate for the anti-ID deliver so far is about 90%. For the blocker, success rate is about 95%. Non-blocker, which is about 90%. As we know that blocker will be more easier to generate compared to the non-blocker, and then we can actually after multiple rounds of counter screening after the total human IgG or like IgG1 or IgG4, the majority of the remaining positive binder are bind to the CDR regions, showing that partial or total blocking effect between the antibody drugs and its ligand. So in summary, anti-ID positive rate to immunogen is very high,
Mhm.
but after counter screening, some majority of them still the blockers. Non-blocker will be more difficult. Yeah.
Okay. Thank you so much for that, we phone. All right, let's go to the next question. Oh, here's it. This is interesting. Among all types of drugs, which types are the easiest to prepare anti-ID antibodies?
Okay, good question. So based
Yeah.
on our experience, and the ADC are actually easier to produce among all other types. Why? Because the conformation of some antibody may change during the payload modification, so which will lead to a greater difference from others antibody. So
Mhm.
therefore, the success rate for the ADC normally is easier and highest.
Okay, all right. Let's go to this next question here, and thank you for answering that. This audience member is asking, what is the price difference between mouse and rabbit monoclonal antibody?
Okay, so for the pricing why, you can actually reach out to our sales manager
Mhm.
from GenScript, and then we can actually discuss further with the project discussion and see what we can actually offer to you.
Okay, and I'm going to share a email address at the closing of this webinar, so please stay with us until the end so I can give you that email address. Okay, let's jump to the next question here.
Mm.
Can the amount of antibodies in animals during the preparation process of ADA reflect the immunogenicity of the drug in humans?
Mm. It cannot because the target immunogen we use for ADA generation is sometimes we will actually add in with adjuvant, and then the administrations of the drug to the human sometimes is different from what we actually immunize in the animal. Therefore, it won't yield the same result. Yeah.
Mhm, mhm. Okay, all right, thank you. Okay, it looks like we have two more questions left. Remember, audience members, you can still send your questions through using that questions window. And for those of you joining us on LinkedIn, please go right ahead and use that comment section, and we'll try to get to your questions during the time we have together. All right, so the next question is how to select polyclonal and monoclonal anti-ID generation in drug discovery development. So how do you
Okay.
select?
So for monoclonal, normally we use to detect the plasma concentrations of antibody drug for PK and ADA assay. So for polyclonal anti-ID normally purified directly from the antiserum, so they
Mhm,
can better simulate the real conditions in the blood sample of the subject. So considering the diversity of the ADA type in the blood sample, and polyclonals are mainly used for immunogens assessments.
okay, sorry. I'm just here grabbing some more questions that are just coming in at the last minute. So give
Mm,
me 1 second as I get them all organized here.
yeah, sure.
Okay, perfect. All right. So the next question I have is what strategies do you use to generate and differentiate blocking and non-blocking anti-IDs?
So for the anti-ID monoclonal discovery, we will actually
Mhm,
first screen the antigens and then see that binding ability, and then afterwards we will perform a counter screenings on the total human IgG and human IgG 1 or 4.
mhm,
So depends on the customer assay application, either they want to use a blockers or non-blocker. We will actually perform a blocking assay with ELISA format, so that the blocking assay is just like a competitive ELISA. For
mhm,
example, we will normally coat the antibody drug or antigens on the plate and add the antigens or antibody drug with our B cell supernatant at the same time, and we do a comparison between our control well, which is like a blank PBS and media inside, and the OD level will be actually different, and we can actually calculate the blocking or competitive ratios of
mhm.
the B cell supernatant. So therefore, we can actually differentiate the blockings and non-blocking anti-ID.
Okay, thank you for that. This looks like it will be our last question. Um, this audience member is asking, how do you ensure that ADA generated against siRNA is specific to the target siRNA sequence?
Okay, so for this one, because we're normally using that for we're using antigen affinity purifications on that, so we
Mhm,
actually using the sRNA to put on the pool. Then afterwards we perform the another rounds of the purification. So therefore, we are specific. Those pulling out is the one which is binding to the sRNA.
okay. All right, perfect. Thank you so much for that, WePhone. Well, thank you very much. We have reached the end of the question and answer portion of the webinar. If we couldn't attend to your questions, the team at GenScript will follow up with you after this presentation. And for those of you who are interested in hearing more from GenScript, you can register for their next webinar, Unlocking CRISPR, Advances in Base Editing, Prime Editing, and Future Applications. And that's scheduled for Wednesday, October the 30th. And I'm going to put the link in the chat box right now just to make it that much easier for you to register. And there you go. It's in there. If you have any further questions, and this is for the person who was asking about the information earlier about the email address, here it is. You can use this one here for WePhone, and she'll be very happy to assist you and to direct you to the right people in regards to
Yes.
that. And if you have any questions at all, just please go ahead and reach out to her. She's a great person to work with. Okay. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Additionally, give me a few seconds. I'm going to share a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Okay, and if you want further resources, I'm going to put this link in here. And let me just get that here. So this link here, you can use for further resources in regards to antibody services. All right, so thank you everyone for participating today. Thank you for your time here today. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. Until next time, please take care and bye for now. Thank you, Wifon. Bye, everyone.
Thank you. Bye, everyone. Thank you.